Alright, in this video we're going to go through some revision notes for Biology 0610 Cambridge IGCSE for topic 6, Plant Nutrition. So let's begin. Of course, plant nutrition, we all know that autotrophic plants, the only source of nutrition for autotrophic plants is photosynthesis. But before jumping to photosynthesis, let's break the word down into two. Photo means light, and synthesis is the building up of complex molecules from simpler substances. So photosynthesis is the synthesis of light, where complex molecules are built up from simpler substances using light. Photosynthesis is the process by which autotrophic plants manufacture carbohydrates from raw materials using energy from light. During photosynthesis, light energy captured from the sun by chlorophyll is used to convert raw materials, carbon dioxide and water, into carbohydrates in the form of glucose and oxygen. So during the process of photosynthesis, chlorophyll, which is present in the chloroplast, captures light energy from the sun, and it uses this light energy to convert raw materials of carbon dioxide and water into carbohydrates in the form of glucose and oxygen. The balanced chemical equation for photosynthesis is 6CO2 plus 6H2O is converted into C6H12O6 plus 6O2 using light energy. The word equation is carbon dioxide plus water is converted into glucose plus oxygen using light captured from chlorophyll. Carbon dioxide diffuses into the leaf through the stomata. Water is taken up by the root and transported throughout the xylem to the leaves in the transpiration stream. Glucose is used to make substances needed by the plant and is also used in respiration to release energy. Oxygen diffuses out of the leaf through the stomata when carbon dioxide diffuses into the leaf, and it's also used in respiration. Chloroplast. Chloroplast is an organelle in the plant cell. Chloroplast contains chlorophyll. Chlorophyll is a green pigment that gives plant their green color and acts as energy transducers to convert light into chemical energy for synthesis of carbohydrates. So chloroplast is an organelle in the plant cell. Chloroplast contains chlorophyll. Chlorophyll is the green pigment. It gives plants their green color, and it converts light energy into chemical energy. Light energy captured from the sun into chemical energy for synthesis of carbs, to build up carbs from simpler molecules. The use, storage, and fate of carbohydrate and glucose molecules. So carbohydrate or glucose molecules, they are used for cellulose for strength in the cell wall. They are converted into starch for storage. They're converted into fats and oils for energy storage in seeds. They're also used in respiration through the mitochondria to release energy for the cell. They're converted into sucrose, which is a type of sugar that is transported around the plant in the phloem, and it combines with nitrates from the soil to form proteins for growth of the plant. Amino acids and proteins. So this is in words. It's converted into starch molecules to act as an effective energy store. It is converted into cellulose to build cell walls of the plant. It's converted into sucrose, a type of sugar which is transported in the phloem. It's converted into lipids, type of fat for energy stores in seeds. It is converted into amino acids to make proteins when combined with nitrogen for growth. It acts as a nectar to attract insects for pollination, and it is used in respiration to provide energy. Alright, what are the mineral ions in chlorophyll? In our syllabus, there are two mineral ions present in chlorophyll, magnesium and nitrogen. Mineral is any naturally occurring inorganic substance. Without these, the plant cannot photosynthesize and grow properly. So plants obtain mineral ions by active transport, absorbed from the soil by root hair cells. Root hair cells absorb mineral ions by active transport. Active transport is the movement of molecules from a low concentration to a high concentration using energy from respiration. This is a illustration showing the deficiencies and needs of nitrogen and magnesium. So in the middle here we have a perfect plant with optimum amount of magnesium and optimum amount of nitrate. On the right side we have a plant with nitrate or nitrogen deficiency. Nitrogen deficiency causes stunned growth because amino acids are needed for growth of the plant, and it causes yellowing in the leaves. While on the left side we have magnesium deficiency. Magnesium deficiency causes yellowing between the veins of the leaves. This is known as chlorosis. So this is the mineral ion in the plant, its function and its deficiency. Magnesium is needed to make chlorophyll. Deficiency of magnesium causes yellowing between the veins of the leaves, known as chlorosis. Nitrate or nitrogen is the source of nitrogen to make amino acids to build up proteins. Deficiency of nitrogen causes stunned growth and yellowing of the leaves. Alright, now we're going to begin the experiments for this chapter. The first experiment is investigating the need for chlorophyll. To investigate the need for chlorophyll, we test the leaf for starch. This test is similar to the test in chapter 4, biochemical food test for starch. Starch is stored in the chloroplast of the plant, not in the chlorophyll. Alright, so the procedure. First, we drop a leaf in boiling water to kill the cells and break down the cell membrane. Then, we leave the leaf for 5 to 10 minutes in hot ethanol boiling tube to remove the chlorophyll and let iodine color change make more clear. So the reason we do this step is to remove the chlorophyll, because removing the chlorophyll will let the iodine color change be more clear. If we don't remove the chlorophyll, we cannot identify the iodine color change, whether it's a positive or negative result. After these two steps, we dip the leaf in water to soften it, and then the leaf is spread out on a white tile covered with iodine solution. Now there are two cases. In a green leaf, the whole green leaf will turn blue-black, as photosynthesis is occurring in all areas because starch is present in all areas. So this is a positive result. In a variegated leaf, only areas with chlorophyll stain blue-black, while areas with no chlorophyll remain orange-brown, which is the color of the iodine solution, as no photosynthesis and no starch is stored. Variegated leaf is a leaf partially green and partially white. So in a variegated leaf, only the green part will stain blue-black because starch is stored there. But in the white part, there is no starch because there is no chlorophyll. So it will remain orange-brown. This is the illustration for this experiment. We dip the leaf in boiling water to kill the cell membranes first. Then we put it in a boiling tube to remove the chlorophyll. And then the leaf is washed in water to soften it. Two notes to remember here is that ethanol is flammable and the Bunsen burner should be off at all times. Of course, after the boiling tube is done. And the safest way to heat ethanol is an electric water bathtub or a thermostatic water bathtub. After the leaf is softened, we place it on a white tile and dip iodine solution to it and observe the color change. Okay, that's the first experiment done. The next experiment is investigating the need for light. The procedure for this one. We destarch a plant first by placing it in dark 48 hours before the experiment to ensure all present starch in leaves will be used up and not affect the experiment. Destarching the plant means we place it in dark 48 hours before the experiment or a specific amount of time before the experiment. This will ensure that all the present starch in the leaves is used up in the plant and it does not affect the experiment in any way. After this, the leaf of plant will be covered partially with aluminum foil and placed in sunlight for a day. So we cover the leaf partially with aluminum foil. only half of the leaf will be covered and the half will remain open. Then we test the leaf for starch using iodine solution. The area of the leaf covered with aluminum foil will be orange-brown as it received no sunlight and could not photosynthesize. The area exposed to sunlight becomes blue-black because it was exposed to sunlight and it did photosynthesize. Alright. The third experiment is investigating the need for carbon dioxide. To investigate the need for carbon dioxide, again, we destarch two plants, here there's two plants, by placing them in dark. The reason we do this is to make sure all the starch is used up in the plant and it does not affect the experiment in any way possible. After this, we place one of the plants in a bell jar containing a beaker of sodium hydroxide to absorb carbon dioxide from the surrounding. So one of the plants is going to be placed in a bell jar next to sodium hydroxide. So this way, the plant will not absorb any carbon dioxide. All the carbon dioxide will go towards sodium hydroxide because sodium hydroxide absorbs CO2. As for the other plant, we place it in a bell jar containing a beaker of water to not absorb carbon dioxide from the surrounding air. So the other plant is also going to be placed in a bell jar, but next to water. This way, water cannot absorb carbon dioxide, of course, so the plant is going to absorb all the carbon dioxide. Then we place both of the plants in bright light for several hours and test both plants for using iodine. The leaf placed near sodium hydroxide will become orange-brown because it couldn't photosynthesize due to lack of CO2. Of course, this leaf, sodium hydroxide, was absorbing all the carbon dioxide, so the leaf itself was not able to absorb carbon dioxide and photosynthesize, so it will remain orange-brown, while the leaf near water will become blue-black because it was able to absorb all the carbon dioxide since water does not absorb any carbon dioxide. This is the illustration of this experiment. So we have plant A and plant B. Both are placed in a bell jar. This is the bell jar or glass container. The first plant is placed near sodium hydroxide, which absorbs CO2. The second plant is placed near water, which does not absorb CO2. What's going to happen is that this plant is going to be orange-brown, and this one will be blue-black. Because this one is able to absorb all the CO2, and in this case, all the CO2 is being absorbed by the sodium hydroxide. So this plant is not able to absorb any CO2. Okay. Now, the second part of experiments in this chapter is investigating the rate of photosynthesis. By rate of photosynthesis, we mean the speed at which photosynthesis occurs. There are two methods to investigate the rate of photosynthesis, and both of them are dependent variables. Just to recall what we mean by dependent variable are all the variables or any variable that can be measured in the experiment. It's any measured variable. These two methods are counting number of bubbles and measuring the oxygen volume. First, counting number of bubbles. Plants and water release oxygen as bubbles, as oxygen is one of the byproducts of photosynthesis. So the number of bubbles produced per minute records the rate. More bubbles produced per minute is equal to faster rate of photosynthesis. So plants are constantly releasing oxygen every time they photosynthesize, and they release oxygen in the form of bubbles. So the faster photosynthesis, the more bubbles produced per minute. The next is measuring oxygen volume. Again, the oxygen produced as bubbles is collected in a test tube inverted over a top of a pondweed over a long time, and we measure the amount of volume of oxygen released to determine photosynthesis. The greater the amount, the greater the rate of photosynthesis. Plants used in these experiments in our syllabus is Elodia and Camboba. To investigate the rate of photosynthesis, we have 3 experiments. The first one is effect of changing light intensity. We move a lamp for different distances away from the beaker with pondweed. So effect of changing light intensity, this means light intensity is the independent variable here. So light intensity should be constantly changing throughout the experiment and everything else should remain constant. Distance and number of bubbles is inversely proportional. If you increase the distance, the number of bubbles will decrease. Inversely proportional. Again, since light intensity is the independent variable, we keep it changing and everything else constant. Let's have a look at this illustration. To measure the light intensity, of course, we use the lamp. The lamp shoots light at the beaker. So we have a scale here. This scale or ruler measures the distance at which we move the lamp. We keep moving the lamp at different distances since this is the independent variable, and we keep everything else controlled or dependent variable. Only change the light intensity. We place a glass tank in between the lamp and beaker to absorb heat from the lamp and avoid changing the water temperature. Because if there is no glass tank in between, water temperature is going to change, and we need to make sure water temperature remains constant because it is not the independent variable. Water temperature in this experiment is the controlled variable. So it must remain constant. The next experiment is effect of changing temperature. Effect of changing temperature means temperature is the independent variable in this experiment. You change the temperature of the water in the beaker. So in this experiment, since temperature is the independent variable, we keep temperature changing and everything else constant to measure the effect of changing temperature. The most bubbles produced is the optimum or fastest rate. So this is the number of bubbles produced of oxygen. The most bubbles produced is the optimum temperature and the fastest rate of photosynthesis. Let's have a look at the illustration. This is to measure the temperature of the beaker. In this scenario, we're keeping the light intensity constant because this is the controlled variable. And we keep changing the temperature because temperature is the variable to be measured in this experiment. Factor being investigated is temperature, so this is the independent variable, must be changed at all times. The third experiment is effect of changing carbon dioxide concentration. Effect of changing carbon dioxide concentration means carbon dioxide is the independent variable in this experiment. So we dissolve different amounts of sodium carbonate in water beaker. And in this, we keep carbon dioxide concentration changing because it's the independent variable, the variable being investigated, and everything else shall remain constant. Let's have a look at the experiment illustration. Light intensity is to be kept constant. Temperature of the water is also to be kept constant, but carbon dioxide concentration is to be changed because it's the independent variable. There are two other ways to measure the oxygen given off. The first one is measuring the gas volume by an inverted measuring cylinder and graduations filled with water, or using a syringe attached by delivery tube to funnel. The three experiments are for light intensity, temperature, and carbon dioxide. Whenever light intensity is being investigated, temperature and carbon dioxide should be constant. Whenever temperature is being investigated, light intensity and carbon dioxide shall be constant. And whenever carbon dioxide is being investigated, temperature and light intensity shall remain constant. The time per minute of bubbles of oxygen produced is always the controlled variable. Alright, now we're going to start investigations and experiments for gases. Aerobic respiration and photosynthesis based off gases. In aerobic respiration, the process is taking in oxygen and releasing carbon dioxide in the plant. It occurs all the time. Plants take in oxygen and excrete carbon dioxide at all times. Photosynthesis is the opposite of aerobic respiration. During photosynthesis, plants take in carbon dioxide and release oxygen, and it only occurs in the daytime because plants only receive sunlight during the daytime, so they're only able to use light energy to convert carbon dioxide and H2O into glucose and oxygen during the daytime. So at night, plants don't photosynthesize, but they only respire. During the day, photosynthesis rate is greater than respiration rate. Net intake of CO2 is greater than net output of oxygen. While during the night, respiration rate is greater than photosynthesis rate. Net intake of O2 is greater than net output of CO2. A short fact, CO2 is an acidic gas when dissolved in water. In this illustration, we have respiration and photosynthesis. Respiration, as you can see, is occurring in both the day and the night, while photosynthesis is only occurring in the daytime. Investigating gas exchange. To investigate the effect of light on a net gas exchange in aquatic plant, aquatic meaning a water plant, we use a pH indicator. The pH indicator used in our syllabus is the hydrocarbonate indicator. This is a color indicator that shows different levels of carbon dioxide concentration in the solution. So this indicator shows different levels of carbon dioxide concentration in the solution. Let's have a look at the table to understand what this means. Here we have the concentration of carbon dioxide, the color of this hydrogen carbonate indicator, and the condition on the plant. The highest is yellow. The higher, but not the highest, is orange. So during these two, carbon dioxide concentration is very high. The color is orange or yellow, and during this time, there's more respiration than photosynthesis. This is a low pH, more acidic. Atmospheric level, this is balanced, red, where photosynthesis is equal to respiration. The lower ones is magenta and purple, where more photosynthesis than respiration. This is a higher pH, more alkaline. So in acidic conditions, where there's a low pH, respiration is greater than photosynthesis, it's yellow or orange in the color. In alkaline conditions, which is in high pH, photosynthesis is greater than respiration, the color is magenta and purple. Atmospheric level, where photosynthesis is equal to respiration, the color is red. This is an example where several leaves of the same plant were placed in a boiling tube with hydrogen carbonate indicator to test the effect of light over hours. These are the results. So we have tube A, B, and C. Tube A and B contain a leaf, while tube C did not contain a leaf. The conditions of tube A was light, B was dark, and C was light. Indicator of tube A turned purple, tube B turned yellow, and tube B turned red. The conclusion of each color. Conclusion of purple tells us that there was a net intake of carbon dioxide by the leaf in the light. Indicate of yellow tells us that there was a net intake of oxygen by the leaf in the dark. And red tells us that it was controlled. Net intake of oxygen was equal to the net output of carbon dioxide and vice versa. And with that, we are done with all the experiments for this chapter. Now we're going to start a new part of this chapter known as limiting factors. Limiting factor is something that is present in the environment in such short supply that it restricts life processes. So it's something that's present in such short supply that doesn't allow the organism to carry out its life processes in an efficient manner. There are three main limiting factors for photosynthesis in our syllabus: temperature, light intensity, and carbon dioxide concentration. A note that water is not a limiting factor because the amount of water needed is very small compared to the amount of water transpired from the plant. So the amount of water that is lost by the plant is very small compared to the amount of water that's actually needed for the plant. The first limiting factor is temperature, which is directly proportional. As the temperature increases, the rate of photosynthesis is also increased, but in temperature, the reaction is controlled by enzymes up to the optimum temperature. After optimum temperature, of course, the enzymes begin to denature and the rate of the reaction decreases. So in this graph, as the temperature is increasing, the rate of photosynthesis is increasing up to optimum temperature, where the rate of photosynthesis is at its optimum. After this, increasing the temperature will cause the enzymes to denature and reduce the rate of photosynthesis. The second limiting factor is light intensity. Light intensity is also directly proportional. The more light a plant receives, the faster the rate of photosynthesis, until some other factor needed for photosynthesis becomes limiting in short supply and prevents the rate from increasing. So, at low light intensities, first, if you increase the light intensity, the photosynthesis rate will increase. It will keep increasing until it reaches a certain point. At a certain point, the light intensity will completely stop increasing because the plant has received enough light and something else will become limiting factor now. This can either be the temperature, maybe not high enough, or not even enough carbon dioxide. But no matter how much light intensity is received by the plant, it's not going to affect the rate of photosynthesis. This is the graph for light intensity. So as we're increasing light intensity, the rate of photosynthesis is increasing up to a certain point. After this point, light intensity stops increasing and some other factor becomes limiting here. As for carbon dioxide concentration, the same rule applies to carbon dioxide concentration, directly proportional. More carbon dioxide present inside the plant, the faster the rate of photosynthesis, until some other factor needed for photosynthesis becomes limiting in short supply and prevents the rate from increasing. So carbon dioxide concentration will keep increasing, and this will increase the rate of photosynthesis until some other factor for the plant is becoming the limiting factor, and the carbon dioxide will not become the limiting factor. This can either be the temperature may not be high enough or not enough light received by the plant. A quick exam tip is in the section where the graph rate is increasing, the line is going up, the linear section, the limiting factor is whatever is labeled on the x bottom axis. While the section of the graph where the rate is not increasing, the horizontal line after the linear part, the limiting factor is any factor other than what's on the x-axis. This is the graph for carbon dioxide concentration. It's the same for light intensity. The last part of this chapter is investigating and discovering the parts of the leaf and their functions. Features of a leaf. Method of gas exchange between leaf and their surroundings, diffusion of carbon dioxide and oxygen, way of delivering water to the leaf via osmosis or transpiration stream up the xylem vessels, system of removal of glucose, and efficient means of absorbing light energy through chlorophyll. The leaf structures present in a plant is chloroplast, cuticle, guard cells, stomata, upper and lower epidermis, palisade mesophyll cell, spongy mesophyll cell, air spaces, and vascular bundles, xylem and phloem. This is a cross-section of a leaf structure. This is the upper epidermis. This part is the upper epidermis. This is the column-shaped palisade mesophyll cell. These are the spongy mesophyll cells. This is the lower epidermis. This is chloroplast present in the palisade mesophyll cell. This is vascular bundles, xylem on top, phloem on the bottom. And this is air spaces in the spongy mesophyll cell. Cross-section of this area. This is the lower epidermis. This is the midrib, and this is the upper epidermis. Midrib is the section separating the upper epidermis from the lower epidermis. Leaf structures and their functions. The first structure is waxy cuticle. Waxy cuticle is a protective layer on top of the leaf, and it prevents water from evaporating. So it's like a waxy layer on top, and this prevents water from evaporating outside the plant. Then we have upper epidermis. Upper epidermis is thin and transparent to allow light to enter the palisade mesophyll cell underneath it. Right underneath the upper epidermis, we have a bunch of palisade mesophyll cells. This is why this is transparent, so light can enter and the plant can photosynthesize. Palisade mesophyll cell is a column-shaped cell tightly packed with chloroplast to absorb more light, maximizing photosynthesis. Spongy mesophyll contains internal air spaces that increases the surface area to volume ratio for diffusion of gases and carbon dioxide. Lower epidermis contains guard cells and stomata. Guard cell absorbs and loses water to open and close the stomata and allow carbon dioxide to diffuse inside and oxygen to diffuse outside. Stomata is where gas exchange takes place. It opens during the day and closing during the night. Evaporation of water also takes place here and is found underside of the leaf to produce or reduce water loss. Vascular bundle contains xylem and phloem to transport substances to and from the leaf. Xylem's function is to transport mineral ions and water by transpiration. Phloem is for translocation of sucrose and amino acids from photosynthesizing to non-photosynthesizing regions in the plant. The adaptations for each feature in the plant. It has a large surface area for the diffusion of carbon dioxide and absorption of light for photosynthesis. It's very thin to allow carbon dioxide to diffuse into the palisade mesophyll cells quickly. It contains chlorophyll to absorb light energy for photosynthesis. It has a network of veins to allow transport of water to cells, leaf carbohydrates from the leaf for photosynthesis, and water for photosynthesis. Carbon dioxide acts as a product of photosynthesis. Stomata allows carbon dioxide to diffuse into the leaf and oxygen to diffuse out. Epidermis and the thin cuticles, they allow light to reach the palisade cells and they protect the leaf without blocking sunlight. Palisade mesophyll cells maximize absorption of light, of course for photosynthesis. Spongy layer has air spaces to allow diffusion of gases, and vascular bundles helps for transportation and support the plant. How do photosynthesizing cells obtain carbon dioxide? The pathway of carbon dioxide from the atmosphere is done by diffusion from high concentration to low concentration. First, carbon dioxide is in the atmosphere, then it goes into the air spaces, from air spaces into the spongy mesophyll tissue, then mesophyll cyst cells, then chloroplast. So this is carbon dioxide, a high concentration of carbon dioxide. Diffusion causes carbon dioxide concentration to go from high to low concentration inside the plant, from atmosphere into the stomata through the guard cells, then into the air spaces, then the spongy mesophyll, then the palisade mesophylls, into the chloroplast and chlorophyll to photosynthesize. The last part of this chapter is an electron micrograph of a plant cell. This is like a cross-section of the plant cell, but taken from an electron micrograph image. This is the spongy mesophyll. This is the lower epidermis. This is the upper epidermis. This is the waxy cuticle. These are column-shaped palisade mesophyll cells. This is the chloroplast. This is the stomata containing guard cells.